Okay, why don't we get started? So good morning, good afternoon, everyone. We joined in the middle of the August. So today, this meeting will be recorded as usual. Today, I'm planning to talk about migrations of our data release manuscript to MIST and collect some of your feedback on this potential migration. Um, And I'll start with a little bit of a rationale as to why I'm proposing we do this. And then I was planning to show you the new things that this enable and we can discuss the way forward based on that. So our data release manuscript has been iterated a lot on this Google Doc document that was shared with everyone. And we've written down, I think, 95% of the methods. We had started to shape a number of figures, and I'm thankful for everyone's contribution there. And we are now working towards the analysis, as well as visualization to help people get onboarded in making the analysis figure the first. And I remind everyone the goal of this manuscript is mostly to introduce people to the data and sort of facilitate any downstream analysis. Uh, what we saw in the last few meetings is that, well, first, agentic AI is just taking off and being used for analysis more and more. And that's one. Second is because this is a data release, it's incredibly helpful to have visualizations to help educate people on the content of the data. And third, because this is a large group that is collaborating disseminated across the world, I felt it will be helpful to have a centralized piece where the entire manuscript lives instead of with the code, because in addition to just the prose. So I have been using Mist for many different other effort, and so I was very familiar with it. And so I took the time to try how it will look. So this is the link that is here. So Mist is just for a brief introduction. Mist is a publication format developed by the Jupyter Book Foundations in Berkeley, as well as many others, but we used for the OpenScope Data Book. They made a newer version in a year or so ago, a new release that greatly extended the standardization of the manuscript. So MIST contain all of the things that a typical manuscript would contain. So a bibliography, and it's based on Markdown. So you write down a paper in Markdown format. So this is a human readable text file that is in on the repository here. But what is powerful is it's very easy to sort of add interactive component on top of this because everything is code essentially instead of just a Word document. So this migration is here, and so I'm just going to go through it, and then please feel free to interrupt if you have any questions. So the first thing is I took the Google Doc and I migrated the content to Markdown using assistance from AI. And so the text that is here should be very familiar. This is exactly what was in the Google Doc. And then I started to consolidate the figures, but where in the Google Doc here. So this is what you see here. And the repository now contain all of the figure sources that were used to generate the figures. So previously we had a Google slide link. So this is the previous set of figures. So you will go to Google slide, and then we would interact on this in this place. But it was I felt it was hard to sort of aggregate different sources. So instead, I started to sort of put all of the source files that I had for all the figures. So I include Illustrator files that were used, as well as any PowerPoint that were used to make some of the figures, and then I added documentation for the different source, as well as the Python that you could use to generate some of the panels. So I tried to organize everything around. And because Mist put everything into a repo, then the entire repository can be a better container of the entire manuscript. And this is powerful because then you can clone the repository locally or have an AI agent do this for you, and then instead of having to sort of pull all the information from different places, it's all in one place. So I felt it was just greatly facilitated this effort. So for instance, the figure one previously was a single panel, and because everything was consolidated, so I could start to sort of uh, put things together in sub-panels. So figure one has changed. Now it's introduced the rationale here, and then this is the schematic of the model multimodal pipeline, and then the panel C is the context of all of those experiments, so for each modalities. So this is the same content, but it's just I sort of iterated it on it so that it's clearer to understand. So all of the figures were migrated, and I consolidated them. And then Mist allow certain things that I think are quite powerful. So previously, we had this figure here that explain how a single session is structured. Um, and Carter went to Geneva in early July, and we struggled to explain the stimulus design. And so I felt that there was an opportunity here to sort of build interactive components to explain the stimulus design. And so this is what was here. And so this is exactly the same canvas. So this session here is the same as this session here. But now you can play and click on different components and play back the session as it was designed to be presented to the animal. You can click on different part of it. You can actually see the zebra movie. So this is a small section of it, or of the receptive field. I'd show on the screen. So I feel like it's helpful to sort of educate people. And Mist allow to have those JavaScript component embedded into the paper, as well as every figure has a static counterpart. So the intent is you can generate PDF files or any fixed form version using the static view. And still, if you go on the web, you will see the full experience of the manuscript. So that's the rationale for loose interactivity figures. Are there any questions at this point, or should I just continue introducing the manuscript? All right, I'll go on. Make sure okay. So figure one and figure two just contain the same thing as before. And then this figure here, we had it before. I sort of consolidated as well. And the sources is there, so it has not changed. Now the methods in the Google Doc was, I mean, taking most of the space, and I think it's sort of impact the reading. And so the entire method is collapsible. So you can access it or just decide to read. And I think that I release manuscript should allow a quick read just to understand the data as a whole, like superficially without going in deeply. And so these interactivity also allow for different types of reads. But the method is there. And so all of the references were transition. And I also sort of started to we had some columns, so this was converted into an interactive. This is very nice. It just had this ability to sort of bring in information through your mouse. So, but overall, the method is the same as it was before, except that you have direct links where you can go. So it's very interactive in that sense. And then we spent the last 2 weeks consolidating this view. Um, so this data set is very rich. It has a multimodal data with multiple sessions, many animals. And so we started to create a sort of synthesis of all of the experiments. And I think Severin today was going to introduce you this figure. Severin, are you able to speak on figure 4?
Yes, I am.
Thank you.
Hi, everyone. So yeah, we did a summary of all the sessions for NeuroPixel, Mesoscope, and SLAP 2. So the colors are the same as you've seen in the other figures. So dark blue is for sensory motor, teal is for standard, brown is for sequence, and gold is for duration. So for each modality, you should have the chronological order of when the session happened, and also in striped and red should be the session that are failing for a reason or another. So we still have to tweak a little bit more this figure and check with Slab 2 if everything is correct there, but like most of the sessions and everything is explained there. So I hope it's self-explanatory and then people can have a feeling of how much data this is representing, basically.
The question for Severin on this representation. Well, the nice thing is, so this is the static view of the synthesis of all the sessions. And if you click on interactive, now is Carter here? Do you want to introduce the tables?
Yeah. So I want to point out that these 3 tables are still they're also available on the public spreadsheet link that we've been talking about for a while now for people to access sessions. So this information is not in general, it's not new to you guys. But we think this really embedded view is super nice for, basically for the manuscript. They might be, don't be concerned if they're slightly out of sync because these have to be updated manually in the manuscript, whereas on the public spreadsheet, those get automatically updated by me. So we have a couple different tables here. So this one is the animals table. It basically just tells you what cohort each mouse was in, and then some basic metadata about their sex, and like genotype, basic things like that. Probably the more important one is the sessions table. And so this is not the full sessions table that you see on the public spreadsheet. This is the sessions summary. And basically that just allows you to get a quick view of seeing what stimulus context and what modality each session was. So, there's little tabs up there that allow you you can filter by modality or by the stimulus context. So this is just a useful view to get sort of an overview of, so right, if you just want SLAP 2 of the duration context, you can see what's available. And then we also have this data access table I made, which is new. This is basically supplemental to the sessions table. It has a lot of the same information, especially the first like 5 or 6 columns. But then we also have the Dandy set ID, the Dandy path, and the Dandy link. Those are on the public spreadsheet, but it's just a more condensed, clearer view here. And then there's a couple of columns that we added which are new. So if you've seen our previous meetings, you might recall that there's a number of intermediate assets that we store in order to produce the NWBs. And for each modality, those intermediate assets are different. All modalities will have this session asset, which is basically the raw recording that comes off of the rig. And then there's a number of other ones. So depending on the modality, like if you were to select Mesoscope, you'll notice those final columns change. For a lot of you, it might not interest you to see the intermediate assets. In general, you probably just want to look at the NWBs on Dandy. But if you wanted to do something more involved with like the spike sorting or cell matching or something, or segmentation, you might want to look at these intermediate assets. And they're stored publicly on S3, so you can, those are the links there. Yeah, so the link will take you to this Quilt viewer, which is just a GUI for S3. As we've talked about in the past, S3 is just basically a public server that stores data for you. So you could actually access this data through a number of different like code APIs. This is just like a convenient GUI to view them. And then you'll notice a few of them, like Jerome, if you were to select NeuroPixels again. You'll notice about the CCF assets, for instance, say internal. These are just assets that for basically technical reasons we haven't publicly released yet. So yeah, that's just going to be some of them. But in general, we try to push all of them to the public S3. I guess I should also note that I will be making an announcement on the forum today that we have most of our glutamate SLAP 2 sessions on Dandy now. Yeah, so that's unrelated to this manuscript issue, but if you're someone who's been waiting for SLAP 2 data, I think we have like 8 or 9 additional sessions that are not pilots on Dandy.
Questions on this to Carter or Severin? Okay. So we're going to continue polishing this view, but it's sort of consolidating at this point. And then the paper unfolds to sort of dive deeper and deeper into each individual data set to provide more contextual information and details. This is the overall summary tables that explain everything that is in there. And then we dive deeper and deeper. So we are currently working on a view that explains the content of the NWB files. This is still ongoing, but we have sort of a placeholder table here. And then I started because Mist allow these really quick iterations with AI agent. I started to make more sophisticated views of the data that could be helpful. And so before any processing is done, you might want to visualize the raw data to understand aspects of it. So if you have never touched it, so we wanted to have a figure here that introduce you to the raw. And so similarly, there's a static and interactive view. So for the neuropixels, you can see all of the probes. And so this is an exemplary profile of a recording. This is raw, so it's not common mode corrected. We are considering adding a toggle here to sort of correct this vertical bars they exist in all NeuroPixel recordings, but it's just to allow you to visualize the raw spike band along with the area on the side. Mesoscope has a similar exemplary data, and you can play. All of the planes are selectable, so you can play an exemplary movie. The contrast can be adjusted, so you can see how the raw data looks more clearly. And then we're doing the same for SLAP 2. This view will likely change. I'm iterating with Made, but similarly, you can play. And then because we want to have, for all figures, we want to have a static version. We are sort of working on this one that will sort of intent again is in the static mode, sort of understand the raw data exemplary raw data for each view. I also started to play with yesterday with visualizing where the recordings are done. And so there's a supplementary figure at the very bottom. It was just pushed. This is very nice. So this is all the probes that were recorded for this data set in the NeuroPixel. And so similarly, there's a static and an interactive view. I think it's possible that this figure will go up and be merged with the data visualization. And you can click on any one of those probe and see the shaft or like select mice. And so if you want to understand how the recordings was done for a given mice, this is really helpful. We actually, we're starting to use this to understand, to make sure that our process is good. So this is a really rich view. Yes, Alex?
I've also looked into the CCF files. I probably shared some code today that I put together. Yep. And so I saw that these wiggly lines, I think they probably happen because the tissue is moving and so on. So I decided to interpolate, have straight lines. I think isn't that something we want to do? Maybe other people might look at that and say, "Hey, wait, how can a NeuroPixel be so wiggly?"
That's right. Good question. So maybe we need to explain it better. So every probe is inserted straight, right, in the brain of the mouse. So it's always a straight line. Or maybe if Severin is she? No, she's not here. What we do is we take the brain. So there's a track with fluorescent protein, but sort of it's not a protein, but a molecule, but allow us to see post hoc the reconstructed track in a 3D rendered version of the brain. And then we take this brain in 3D and morph it back to a common coordinate framework. And so that process actually sort of do a transformation that make the straight line wiggle, because and not every brain is the same. And so in some ways, this wiggly line is actually accurate locations of the probe in the shared template. Does that make sense? Because all the brain areas move around a little bit when we realign to the shared template. But it's possible that this process could have, could introduce some wiggles a little bit. I would say it's fair, but so far I think we've kept this tracks. Yep. But I mean, any validations of the error assignment would be very helpful. Like if you do any work to sort of dive deeper into this, we could add figures here and to better understand the area that we recorded from. Does that answer your question, Alex? Okay. So we're here, and so this is the raw view, and I've left these figures that few people were working on. So the unit extractions and the SNR and the analysis of zebra, I've left them as they were, and so I just expect those people working on it to sort of contribute their panels here. And so they're here. And there was a figure that we didn't have a direct plan. We said we'll do it later. And so I thought I just gave it a go to try out how to visualize the behavior data. So you might recollect this was in the plan in the raw manuscript. Letter here. Just want to bring it up. Yeah, behavior tracking across modalities. We didn't have any sort of vision on it. And I wanted something that would convey the richness of how the behavior track so that people understand what's possible. And so very similarly, there's two view, a static and interactive view. The static is showing you all of the video feeds that were recorded during this experiment. So at minimum, 3 cameras. The mesoscope had a 4th camera on the nose, a close-up. And what you see on the right is just the simplest things we could look at, which is the running trace throughout the stimulus. So each one of this block is the stimulus block. And so these are exemplary running of this mouse on the left. And then the bottom, the reason I did this is just illustrate how we can do analysis across the entire data. And so I wanted to illustrate that process in the manuscript GitHub repository. And so this panel in D is the average running across every block of the stimulus for each modality. So you might notice immediately that in our data, the mesoscope mice on average had a higher running rate than the others. So it's just part of the data we collected. So this and to generate these panel, there's code in the repository that first run a script across all the NWB files, create an intermediary JSON that store with extracted values, and then create the figure from that. So we have provenance of how the figure was generated. And so I did this so that we have some kind of process for making more and more of those figures across the entirety of the data. So that's good. And then because our data is on S3, all of the, yes, Alex?
So related question. So I saw to my delight that there's more and more data coming. It almost doubled. I looked like 2 weeks ago and it almost doubled. So
Yes.
This one, it looked like it's not all the data right now. So is the idea that, so there's only, for example, 3 SLAP sessions and only 8 mesoscope session. So is the idea that before we submit to somewhere, this gets updated to the full data set, or do you want to keep it at the like these numbers? What's the idea?
I think n equal 8. Yeah, we should do this across the entire data set. There is a script that is run that should be rerun. Like, yeah, I think at the very end. I think we should make all of those figures, and then at the very end, when we finalize the manuscript, it should be a rerun to make sure it's current, and we all agree. That's right.
Okay. Related question. So with the SLAP 2, we have these initial 8 sessions or so where we didn't run the, for example, the sensory motor yet, but they're still good sessions. So
that's right.
Do you want to restrict to only the sessions that we have across the 3 modalities where it's the exact same paradigm, more or less? Or shall we also add these sessions, specifically in the SLAP 2, where we're still modifying the paradigm and the order of presentation and things?
We could add it to the C panel. It's just going to be more documentation burden. Because right now this is in like a clean world where everything is matching. But we could add it to the panel C.
It would help because the power goes up, right? So for example, just for the tuning and the receptive field mapping, those are still really nice sessions. And we did some kind of oddball. So these are still might be still interesting to make part of the package.
We could definitely improve the increase here. I mean, we are meeting this week with the SLAP2 team to sort of discuss how we want to display C, because I mean, we need to acknowledge SLAP2 is a much more cutting edge platform, and so we had a little more diversity and technical issues to handle. And so whereas the mesoscope and NeuroPixel was like well established, rock solid, and so we could go all in. And so that is reflected here. But I feel like it's a matter of documentation. Okay, and then this interactive view is so because our movies are on S3, we can stream their behavior videos directly where the data lives, and I wanted something that just build an intuition, allow people to build an intuition about those videos. And so these is a feed from the NeuroPixel behavior camera on the side. Remember, they are free. And then the stimulus that is displayed on the screen is synced up on the right. And so if you click play, um, those are matched to each other, and so the mouse is still, so it's not impacting anything. But depending on the experiment you're looking at, you're going to get different behaviors. So if you look at the mesoscope example, so it has to first stream the data, and then in here, hopefully that works. That's right. So this is a sensory motor coupling a portion where the mouse is controlling the the stimulus. And so as it works faster, the stimulus on the right accelerate. I hope that the streaming was fine. And there were an instance of oddballs that was presented in the middle at the beginning of the movies. You might have noticed. So this is walking forward, and then suddenly an oddball occurs, and then you can sort of build an intuition as to what happened. What's nice is now you can trigger the eye and then see the same thing. And so you can look at the pupil, like, does anything happens when the oddball occurs? And in this case, it stays steady. There are different types of responses that you will need analysis. And so for all of those modalities, you can do this visualization. Oh, interesting. That worked. Maybe need to a refresh. I don't know. Yeah. Anyhow, so this is just sort of the interactive view of this behavior data. Yeah, Karim, there are examples. I invite you to look at the viewer. There are instances of oddball where the pupil changes. You'll see it, but not always. That's right. I mean, it's going to require a lot of analysis.
I can envision,
then,
because these are not easy to I mean, they can be detected, but time locking to the exact, timestamp in the neural data is important.
Yeah.
And the oddball. Yeah.
So in the text, the text is sort of inviting you all to sort of start diving into this data. I think it's going to require a fair amount of processing to do this right. But I mean, we could add more in the data release paper, but I would rather sort of document it well, package it well, and then I think just a follow-up study could be written on the analysis of the behavior data. I mean, you have 10 video feeds across 3 modalities. Yeah, this is total number of mice is in the 30. So there's a fair amount of data.
Yeah, good sample size. Yeah.
Okay, and I don't think I have more new view. Oh, we made this one with Ryan. So this is just using the unit yield. But are there questions on this missed transition generally?
So my first impression, it's great. I think it makes things a lot easy for the people who have contributed, and also the readers. So yeah, great.
So thank you, Karim. There is a piece I did not discuss yet is the authorship. And we're also innovative here. And so this little viewer is something that I've developed with Carter and Dan Bierman in the last few months. So you might notice faces. So those are the current authors that are recorded into a portal. And I'm going to show you this. So what I envision is a very transparent process for people to say, I contributed to this piece. And so the way it works is if you think you contributed a part of this manuscript, you go here on this link, and if this is my view of it, but it will ask you for your ORCID, and then you're going to be logged into a very small portal that allowed to document contributions. And so you enter information and say what you did, and then these can be synced with the manuscript viewer here that is in the Explore button. And so when you click on this, it shows you the credit table. So it shows you who did what. You can actually navigate and see which section a particular person contributed to. This is a draft, so this information still needs to be completely finalized. And you can see how people were related to one another, like which part they contributed to, as well as the usual sorted list. And so this is important because we have so many people that contributed to this piece that providing more transparency and more visualization, I think will help credit the right people. So, and so if you want to contribute to this piece, I think, well, first you should clone the repository that is here. I wrote a little instruction yesterday with AI to help this process. So you go, the README reference here, so it's explained how this will work. And there's a little contributing.markdown file that explain how to best do this, in addition to how to use AI agents. So because this whole thing is packaged in one repository, it's extremely efficient to sort of work with agents, sort of make new figures, integrate, or like get it to go through a lot of material because it has access to the entirety of the metadata. It knows where the data lives. So it's actually very efficient. And so if you want to contribute, I invite you to sort of first clone the repository and then make your explorations or your new plots, finalize it, make sure you validate it, and then you make a pull request on the repository that contain your figures, the new text, or things like that, and then we'll review it. And if it passed, you'll be in the main manuscript. Does it make sense?
Yes, does make perfect sense. I do have a question regarding analysis of this data that has not been either following this format or pulled. It has been ongoing, but then the timing for presenting it to the group wasn't really, made explicit. Where do we go about that? Or how do we go about that?
You mean to convert analysis to go in there? What's your point?
Well, let's say, for example, Nicholas has done some analysis for the validation of receptive fields, and things like that. He created some plots, didn't present them yet to the group. So how do we go about including this for review?
I see. I mean, I would advocate for making so it's explained in contribution here. I think it's important that the figures that are in this manuscript can be reproduced because the data lives on S3 buckets. And so if you go here to the instructions, I would let Nicholas make a pull request. It's explained here. So you just put your figures into this place with the code that generated from the files, and then we'll review it and we can merge it in. Where it will go in the manuscript, I think it's just like a joint decision to make, either a main figure or a supplemental figures. I think it depends on how the story tied together. Does that make sense?
Yes, absolutely.
Alex?
Well, excuse me, it's related to Kareem's question. So first of all, I think this is ingenious, and I have also experience with MyST. I've been working with it for the last year. So great. The only issue is with Google Docs, had this ability to comment, right? And so now we don't. So let's say I saw the abstract isn't written yet. And so
It's not.
somebody takes a first step at the abstract. Well, rather than me just rewriting the whole abstract and sending you a PR where half of it you like and half of it you don't, I can't just comment and say, how about this? And we, then other people chime in and we come to a conclusion. It seems that now that that might be an issue, that basically all you can do is now reject or accept. And so,
No. So you can well, I appreciate Google Doc has a very nice commenting infrastructure and suggestion mode. I think that's what you're leading to. I agree, but Mist is limited there. I actually reach out to the Mist team because they want to build a publication format that allows these things, and so they're aware of its limitation. In the interim, you can still, read the markdown here. You can edit it by hand and then and make a commit, but other people can see. And so I feel like it could still function. Like you could still see the current abstract, edit it directly here, and submit a commit, and then we review it as a group. The nice thing here is the versioning is much better than Google Doc because Google Doc, as soon as you start to collide, it gets complicated. And so I feel like the pull request is a bit of software engineering nerdiness, but I feel like it still function here. But do you think that could work, Alex, or do you think we need more?
Well, it seems to me that at least for the figures, I mean, we're kind of working on, let's say we have this figure about the receptive fields, right? And across the different techniques. And so where different people work on different Python notebooks and so on. It seems that before we have the final code and the final figure, which is, as you said, it would be great to make it part of this repo and have it interactive in Mist, that until we reach that point, we still have these drafts, right? So where you had, okay, here we would like to have that and we haven't done it yet and we fill out. It seems that at least maybe until this stage, we should use Google Slides or some of these other techniques. And then maybe when things are far along, then you directly editing the markup file might be. Because otherwise, I can see with the Google Docs, what was great is that a lot of people were constantly working on it and adding edits and resolving edits and comments. So I understand the version control is better here, but I mean, what are you gonna do if you get 100 PRs a day? So not that there's, yeah.
I mean, we kind of have you want to keep a version of the manuscript in Google Doc, is that right? Or
I don't know. I'm just bringing up the issue that as much as I love all of that, I think that the ease of being collaborative about this. So, for example, let's start writing the abstract here, and it's not as easy anymore as just adding a comment and saying, "Hey, maybe we should drop this word." Now I actually have to set my own IDE up, and so there's already like a technical burden here, right? So I have to clone the repo. You need to know how to do it. You have to set up your IDE, maybe even get agents to understand and do it with agents, and then send you a PR just for dropping a word. And so each time I do it, I send you a new PR, so it's going to be PR after PR after PR. So that's the only downside that I see. Where I'm wondering if we maybe should leave this up maybe until things are pretty far along, at least for the figures. Maybe that we're not quite there yet for some of the figures that we're collectively working on.
I mean, I think we can certainly continue to use Google Doc or like other forms of exchange. There's a tension here because, like all of this interactivity and the use of agents is, I mean, is much easier with GitHub repository. And so like, I feel like on figures, I might push back because I think it's, for me, it's now much faster to have this on GitHub because it can access the data, it can interactively correct mistakes.
Right, but
often we have for the abstract, I think you might be right.
Jerome, we have these multi-panel figures, right? So for example, you have the 3 techniques, the 3 modalities and receptive fields, and somebody just might work on the neuropixels. So
Yeah.
yeah, that is easy then to incorporate here. But how do you make figure 4 where you have these multiple panels and everybody has a different notebook for a different subpanel?
Yeah.
So
Yeah.
I love this for the final step, but I'm wondering as we're still some of the things are not quite as
Oh yeah,
far
but it's
along
fine. I
yet.
mean, I think it's perfectly fine if you guys want to sort of use like iterate the figures quickly and then and at the end we just merge it this way. I think that's fine with me. Davis, do you have comments? Here. I can't hear you. I saw you on mute, but I'm not sure. Okay, maybe technical issues. Yeah, I mean, overall, I feel like what we are missing to finish this paper is the abstract in the beginning. I think the rationale could be a little tied up. And then I think with the institute folks, we're going to finish all of those figures until the data is well explained. And so we can do that. I think mostly it's the unit and the zebra, and then some of a simple oddball response figure. And if those threes are done, I think we'll be pretty close to have the manuscript ready. And then now that the data is sort of almost fully packaged, I think it should be easier. Uh, I think you should see that with this source of information, it's going to be faster to sort of consolidate those figures.
So Dave pointed out that you can have comments on PRs, right? On the GUI.
You can.
But it's just yeah, I mean I kind of would share the concern, right? That it's hard to compare across comments and changes, right? You can only compare across that one PR. But
Yeah.
yeah.
Your questions, Alex, I actually asked it to the MIS people directly last week because I think they want to enable that vision of like a paper that is completely contained with the code, and they're fully aware of this friction that software engineering brings in.
So I had a brainstorm, right? I wonder, so you managed for the authors to have an interactive
Yeah.
thing in here. I wonder, and so I've been using Jupyter Book, which is it's a Mist version. It's not Mist itself, so I don't know what Mist can do. Mist can do more than Jupyter Books, but I'm used to Jupyter Books. But so what you can do there, it seems. So long story short, I wonder if you can have every couple of paragraphs in here some kind of box that anybody can click and leave some comments, and it saves that somewhere, so, kind of like you do for the authorship. So you don't have to clone the repo. You don't have to submit commits. You could just have a feed where somebody says, "Man, I saw a typo in your figure legend."
Yeah. Yeah. I actually tried to look into this a little bit. That the issue is how much time we dedicate to build sort of manuscript infrastructure but the issue is it's possible with JavaScript to do this, and I could get an AI to sort of go crazy and do it. There's an authentication aspect because so when you put a comment, you're sort of putting your name, and you have to authenticate to a platform, and to do this right, I will need to sort of hold on to your authentication token. I need a subserver. I would rather let the MIST people build this than us working on the data release. But I feel like they are committed to doing this, so I feel like we should just help them, the MIST people, just figure out the right way to implement these needs. Yeah. Maybe. I don't know. If you guys want to try to build it, I'm super supportive. Yeah. But at the very least, I feel like the interactive components are, for me, they really help educate. Because this is a data release, so we want to explain how the data was collected. And so without the interactivity, it's very hard. And so I think we should think about, different views of the data that are helpful to onboard.
I mean, the last idea that I have is because Mist makes it easy to, as you said, for example, just turn this into a PDF. It's kind of like LaTeX, right? So you have, instead of LaTeX, you have markup here, but you can turn it into a PDF. Whether, at least at some step, we turn it into a PDF and we let people just comment on the PDF or something. Just because it's been my experience that, what if there are small typos? And so is it really worth for me to clone the whole repo and send you a PR just to fix one typo? Whereas if somebody says, here's a PDF, do you have 10 minutes? Can you go over that? Might be easier. Yeah. So
But have you tried to correct a typo through GitHub? I mean, it's not that, it's just 2 clicks away, but
yeah, I mean, so you have to. So I think for me, because I'm used to Mist, I think it might be easier for a lot of people. So you clone the repo, but if you're not used to markup or you don't know the Mist markup commands, it's
Yeah.
almost like you have to learn a whole nother language, right? So so it's not entirely non-trivial that if I spot a typo here on your, this is rendered HTML, right? And so if I see
Yes.
something here in your HTML, to then find that in the markup. Yeah,
Yeah,
I
that's
don't
right.
want to
And then you have to-
exaggerate, but it's more effort than a Google Doc. I mean, that's all I'm saying.
I agree. I agree with you. I mean, you have to click on the edit button here. And then find the place you want to make the edits. Change the text. Say, that's right. And you commit. So that's assuming you go into the main.
That was now without cloning the repo, right? So you're saying that all of us can go to this original repo and send you a commit, and then you can decide. Okay.
I might even suggest, like, if you're somebody who wants to make changes, I mean, you could just have a PR, right? And then basically those changes would be pending, and you could continue the dialogue of your changes before they get integrated, right? So, like, if you have a series of different changes, the whole dialogue could happen on your PR, right?
I mean, or you go, you create a branch. You say, yeah, but I mean, you say new branch, Alex Meyer changes, create new branch, and now you're into your edits, and then you make your change to the files. You say, oh, I want to change the index, and then you just change it.
Okay, so if I have a
I know
one,
it's
if
not
I have one,
as easy.
if I have one edit, I do not have to create a branch. I just could edit your markup and send you a commit, and you can decide. But if I have multiple changes, I should make a new branch, and you can decide to merge or.
That's right. I mean, if it's a big change, I think it should be approved.
Okay.
But like, if you want, a specific person to review that, you could tag them in your PR, right? Ask them to take a look at, have a dialogue about the suggestions and things like that.
Nicholas, did you have a comment or?
Not relevant to what's currently being discussed, but I did want to see whether or not I can present a receptive field analysis I did of the OFIS mesoscope data, which was done to validate that data set. I could post currently the results that I have onto the GitHub discussion section, and perhaps I can present those results next week, and also submit a pull request potentially.
Yeah, you could try it, see how it fits. next week, next week I was hoping we do a round. So we go through the figures and just see where everyone is at. This week a number of people were out, so we couldn't do it. But I feel like next week that might work. And so if
Okay.
you could present your receptive field analysis, that would be great. If you want to make a PR on the repo, please do so. It's a public repository.
Okay, sounds good. And I'll also post what I've done with a description of the figures and why I think they validate the data to the discussion section as well.
Yes.
So I was part of a hackathon that was sponsored by Anthropic, and so they paid me a lot of tokens, which was nice. It was run by Cerebral Valley, and I used it to analyze the data. I'm gonna post my repo. I started from scratch, do the receptive fields again, so at least we have now different agents always coming to the same result. So I think that is nice. Might be something you want to discuss, but also I actually did all the oddballs also. So there's a lot of, you said maybe we should have that information. There is something that is the same across all techniques, so that might also be interesting to be looked at.
Yeah, great.
It'd be interesting to compare. Yeah.
We need to let's discuss all of this analysis next week. Like let's do a round and just everybody say where they are. We do need a figure on we committed to do figures on receptive field and a figure on oddballs, and those are still draft, and so we need to make progress there.
What about orientation tuning?
We could have it. Yep.
My
Makes sense because the
field figures are relevant to orientation tuning, I think.
Yeah, I think we need that. Yeah. In fact, orientation tuning and direction tuning, to be perfectly clear, right? So we have static stimuli and drifting gratings, but it's relevant to interpret the oddballs.
Yes.
And this is a massive amount of data.
But the figures can be simple. The figures can be simple. For example, you just have some polar plots for each of the techniques, some examples, nice tuning curves, right? So just so somebody knows, here's the code and we do see tuning.
So I have a question. So do we need, are the kind of figures you're looking for figures that are, let's say, specific to an individual session, or do we also want figures that summarize across multiple sessions, across multiple subjects, or both? Do we want all the above?
I think both. Typically, you choose an exemplary session to explain your metric, and then you screen it across. That's what I would recommend. Similarly to how this was done here, the static figure. So exemplary running, then metrics across all technique.
You said, I missed what you said earlier. You said screening across, like there was something I missed.
Let's say you sort of build an analysis on one session, or for example, orientation tuning in the different context, and then you show that analysis for one session in a subset of your figure, and then you build the statistics across all sessions. So to support your finding. And so the typical panel that I will see are like that, where you explain your analysis in one, and then you show its effect across many. Oh, lost Nicholas.
I heard what was said, so I'll communicate that to him.
Okay. Well, I think we've achieved what we wanted to do. So next week we'll discuss a roundtable. Thank you.